with one of your researches, like the populations of the northern and the southern, they have an ancestral lineage which is very grossly different. Can you tell me something about this discovery?
Call these two groups as a Dravidians and Indo-Europeans.
Dr. K. Thangaraj is a distinguished Indian geneticist whose pioneering research has fundamentally reshaped the understanding of human origins, population diversity, and disease genetics in South Asia. Former director at the Center for DNA Fingerprinting and Diagnostics, CDFD, in Hyderabad and formerly a renowned scientist at the Center for Cellular and Molecular Biology, CCMB, he has spent nearly 3 decades exploring how genomic variation reflects the ancient and complex demographic history of Indian populations. His landmark study uncovered that the indigenous tribal populations of the Andaman Islands providing crucial evidence linking India to the earliest phase of global human dispersal. Published in Science in 2005. In 2009, his Nature paper revealed that nearly all contemporary Indians descend from 2 ancient ancestral lineages, the Ancestral North Indians and the Ancestral South Indians. American Journal of Human Genetics in 2013, Nature Genetics in 2017 are some of his important publications.
We have been working in this field for the last 3 decades. So our research journey in this aspect started middle of 90s, looking at various populations. And one of the populations is the Andaman tribes. By looking at their phenotypic features, people, those who are seeing them for the very first time, they would see that how did they arrive the small islands like this. So they are the first modern human who migrated out of Africa through southern coastal route and reached the islands about 65,000 years ago. Some entered into southern part of India, some went to Andaman and Nicobar Islands, some proceeded to Australia. Not only the biologists, not only the scientists, even the common people is very interested in knowing about the history of human populations. Similarly, understanding our health and disease is also very, very important. Fortunately, because I could work on both aspects, I could also mix both for better understanding of our health and disease. The population history helped us in better understanding of some of the rare diseases. Why rare diseases are very common, how it actually happened. We could predict using a genetic tool, and we can actually look at, because we don't know some of the populations whether they have the disease or not. By looking at the genetics, we went back to the population and in fact we found. So
Dr. Thangaraj has received many national and international honors, including the Sun Pharma Research Award 2019 and the Legend of Hyderabad Award 2024.
So very simple example which everybody can understand is our skin color. If you go to Africa, they are all dark. Right? If you go to Europe, China, other places, you will see mostly they're all with a very light skin color. Right? So these two have mixed, right? Like a paint mixing with a different proportion, we'll get different colors. Right? That's the way our genome is mixed. The people who are living in India, you cannot say that she is very pure, not admixed for thousands of years, except Andamanis.
Parts of western pockets,
Right.
do they have different lineages, or it is majorly the same?
If you look at Western India, there was large number of migration took place subsequently. For example, if you take Jewish, they're more in Mumbai, also in Kerala. Then Siddhis, they were in very small number in Gujarat, Karnataka, and also they are in Hyderabad. And that each time when the people migrate, there's a local admixture. So today, if you take a small village, all the individual, all the family must have come from a common ancestor. That's a living. Then we extend it further, worldwide, ultimately everybody will go trace their origin, go to Africa. Okay.
Why did you select mitochondria?
So while we inherit equally the nuclear genome from both the parents, we inherit mitochondria only from the mother. So we want to trace evolutionary fast because every generation we inherit off from father or from the mother. After a few generations, very difficult to actually to trace the lineage. Using the mitochondrial DNA, because it goes generation after generation without much change, you can trace the maternal lineage in much better way.
These days we come in the news, we see, at a heart attack at a much younger age. So can genetic studies help to identify that this person is predisposed to having a heart attacks?
Yeah, not only heart attack, any disease can be predicted. Some of the diseases, including the cardiomyopathy, can be delayed.
How consanguinity is practiced in Indian communities? Does it have health effects?
Not only the consanguinity, and also the endogamy is also causing larger number of recessive disease. So in fact, we predicted that at least 1/3 of the Indian populations are expected to have such a disease.
So, sir, I'll start with the very fundamental, because one of your research is like the populations of the northern and the southern, they have an ancestral lineage which is very grossly different. So, how did you come to this? Can you tell me something about this discovery?
Thank you for coming and asking these questions. So not only you, there are many common people who are interested in knowing about our ancestors. Yeah. You may be aware that we have been working in this field for the last 3 decades. Right. So our research journey in this aspect started maybe in middle of 90s, looking at various populations. And one of the populations is the Andaman tribes. Yeah. By looking at their phenotypic features, people, those who are seeing them for the very first time, they would see that how did they arrive the small islands like this? Like that. It's also very interesting for us to go and collect samples from them. Right? And then we looked at their DNA, although phenotypically they are similar to Africans, but the DNA variations are different from the Africans. And then when we investigated using large number of markers, and we found that so they are the first modern human who migrated out of Africa, okay, through southern coastal route and reached the islands about 65,000 years ago. I mean, they are in isolation for the last 65,000 years.
Wow.
And this is the period, whatever the mutations they have acquired makes them very unique. Okay, in that process, many things had happened. Some group stayed in southern part of India. Some moved further to Australia. Right? That means the first wave of migration which took place from Africa through southern coastal route, some entered into southern part of India, some went to Andaman and Nicobar Island, some proceeded to Australia. So this is the very first travel the modern human took. Those who settled in southern part of India, whom we call as Ancestral South Indians.
Okay.
Right? Then there was a second wave of migration from Africa, went towards north. Maybe somewhere in the Middle East, there was a divergence. One group has gone to Europe, other group has come to northern part of India, whom we call as Ancestral North Indians. Right? So these two are the founding groups. Although we have, we call these two groups as a Dravidians and Indo-European, there are two more linguistic groups, Austroasiatic and Tibeto-Burman, who must have migrated later on. So we are not really talking about those two. If you talk about the founding populations, these are the two founding populations, Ancestral South India and Ancestral North. And you can also imagine that somebody settled in the very jungle 65,000 years back, or even more than that, 70,000 years. Nothing would have available except the jungle, right? People who stayed there, and they must have finished all the food available there. And those who are active to explore other places where they can find more food, they must have migrated to other places.
Basically, the resources have become limited.
So people migrated to other place in search for food. then from that to other places. That's how the human migration must have started to moving outside the place where they have settled for the first time. To the happen on both the sides. From south, they must have migrated towards north. From north, they must have migrated towards south. Okay, right? So this happened uh, until 4,000 years back. Then during the last 2 to 4,000 years, there was a mixing of these two, ancestral South Indian and ancestral North Indians. Right? So very simple example which everybody can understand is our skin color. Okay, right? Right. If you go to Africa, the founding Africans, very first modern human, they are all dark. Right? If you go to Europe, China, other places, you will see mostly they're all with a very light skin color. Right. That is again, there is a evolutionary pressure which made their skin color into lighter. We are not going into that detail. So these two have mixed, right? Like a paint mixing with a different proportion. We'll get different colors, right? Right. That's the way our genome has mixed and gave so many shades of skin color. You have extreme dark, you have extreme light. In between, there are many colors. Right, right. So that's a very simple example where people can understand, yes, we're all admixed. Today, the people who are living in India, you cannot say that she is very pure, not admixed for thousands of years, except Andamanis. All other populations are mixed. We can actually see that in the DNA, that how much of proportion of the South Indian has North Indian genome. Same way how much of North Indian have the South Indian. I'm talking about ancestral North. Yeah, not the person. So that gives actually very interesting aspect of the unity, that we're all admixed. Nothing is very special either side.
Either side, right? But sir, this is very curious to know in Andaman population how they manage to have not having admixtures within them and retain their gene pool from their ancestors for such a long time.
So they settled, actually, there are 4 tribes. Greek Andamanis, Onge, Jarvad, Sentinels. Right. Each one of them living in different islands.
And isolated.
Yeah, they are all isolated.
Okay.
Okay. Even within these two groups, there is no admixture. Because they have moved to a particular island, they remain there. Right? So there was no mixture. But originally, if you look at the Y chromosome and mitochondria, they have their original kind of M31, M32 haplogroup. Right? Only the Great Andamanis have lost their original Y. They have admixed very recently. Otherwise, these 3 groups, Onge, Jarva, and Sentinelese, are with their own, although Sentinelese has not been analyzed so far because they are in isolation. The American go near.
Yes. Right. Yeah, sometime back one person tried to enter.
Yes,
That
yes.
incident happened.
Yeah, so that happens. So
So if today also we try get their sample, blood
Yeah.
sample,
Yeah.
I say we can find out something more.
absolutely, absolutely. Yes. Yeah.
Great, great. So, again, already my next thing was like how this Andaman population and all, which you brought out already. But then, when we are talking of out of Africa theory, out of Africa theory has many versions of it, right? And with your work, I think in Indian population, we have got a very firm baseline that how this happened, which you beautifully explained it. Now, again, is it possible that grossly, if I take the entire North Indian population, they have the similar lineage and the Southern India, or there are pockets which might also differ? Like, for example, like, parts of Western pockets, like the Kutch area or the West Bengal, uh, the eastern part of India.
Right.
Do they have different lineages, or it is majorly the same?
So majorly it's the same, but if you look at Western India, there was large number of migration took place subsequently. For example, if you take Jewish, they're more in Mumbai, also in Kerala. Right? Then Siddhis, they were in a very small number in Gujarat, Karnataka, and also they are in Hyderabad. And Portuguese due to trade. There are many migrations. Yeah. And that each time when the people migrate, there's a local admixture. Correct. Right? And you can see that in some of the local people, some of the West Eurasian markers, right? Same way in the Jewish, you can see Indian markers or Indian genome, and putting it a simple way for people to understand. So these are all happening in the recent past. But otherwise, look at broadly the majority of the populations. It's kind of admixture. Even if you take the Parsis or although small admixture Parsis, Jewish, Siddhis, and many more population have migrated recently. So everybody has admixed. There's none you can say they are very pure or something.
Yeah, I think that is where some people who have that, no, I am of a pure class and all, their, I wouldn't say the word ego, but their pride might get hurt at some time when they know about this. But it is more about see, one
Well,
aspect
it is, so in this case, actually, one has to think broadly, right? So today, if you say that there's one family, and they can say that they are from this particular family, right? Right. If you take a small village, all the family must have come from a common ancestor. That's a leap. Then if we extend it further at district level or state level, right? And worldwide, ultimately everybody will go trace their origin, go to Africa. Africa. Okay. So how many hundreds of years, thousands of years back this happens once you have that kind of broad idea, then all these fightings will
cannot happen. I think it's very nicely said. We all have branched out from the same area,
correct.
same source.
Correct.
So there is, if you see, holistically, we are actually of a larger same family.
Correct.
Right. Yeah. True. Sir, then for to understand as a geneticist, population geneticist precisely, why did you select mitochondria?
So mitochondria, I think you are well aware, we all inherit mitochondria only from the mother. Correct. Right. Uh, Irrespective of male or female. There's no paternal contribution, right? Uh, for evolutionary studies, if you take small number of markers from the nuclear genome, like the chromosome, we have 46 chromosomes. Right. 23 we inherit from mother in the form of egg, 23 from the father in the form of sperm. It fuse and become again 23 pairs or 46 chromosomes. Correct. That's the reason we inherit the all the phenotypic features equally from both the parents. Sometimes whichever dominant gene, so that kind of features the child will have. So while we inherit equally the nuclear genome from both the parents, we inherit mitochondria only from the mother. Yeah, right? So we want to trace evolutionary fast because every generation we inherit off from father or from the mother. After a few generations, very difficult to actually to trace the lineage. It use very small number of markers, but now we use whole genome that gives more opportunity. But when we have limited resources, limited number of markers, we cannot really use the nuclear genome to trace the origin because every time we are inheriting off of it from both of them. Whereas the mitochondria goes as such from the mother to son or daughter, again son cannot transmit to the next generation. So daughter transmit. So using the mitochondrial DNA, because it goes generation after generation without much change, you can trace the maternal lineage in much better way. Same way, if you want to trace paternal lineage, all the males will have Y chromosome, one Y chromosome, right? Right? Female doesn't. So if you want to trace the paternal lineage, because again the Y chromosome goes generation after generation without much change in the male lineage.
Right.
Grandfather to father, father to son, then grandson, it goes without much change.
Correct.
So if you want to trace paternal lineage, we use Y chromosome markers. So that's the reason we use both mitochondrial DNA and the Y chromosome DNA for the evolutionary studies.
Right. Right. And if a person does only mitochondrial DNA lineage tracking, not the Y, the story is half. Or do we need to do both to see the sync how it is happening?
So if you do both, it will be good. If you use even the autosomal marker, now there's a possibility of using the whole genome.
Right.
It will give much more information, not only evolutionary aspect, also to understand our health,
Yes.
right? So that's advantage of using it. But in the previous days, maybe we take uh, 10 or 20 years back, uh, very difficult to use the whole genome uh, for both the reasons. One, you need to have enough of fun to get through the whole genome, carry on, right? Second, the capability of analysis and so on, right? That's why now we are in a better position to use the whole genome to understand our evolution in past. If you are using mitochondrial DNA, you can say that we have completely looked at the maternal lineage.
Yeah.
To use the Y chromosome, you can say that we looked at the paternal lineage. But if you use the whole genome, you can use the nuclear genome, you can also use mitochondrial DNA, and of course, the nuclear genome also will have Y chromosome. So that's a complete.
So I think that with the more resources and more, I think, improved technology also,
Correct.
GWAS is what we like genome wide associated sequences has played an important role.
Yes. So genome-wide markers, which after the human genome was sequenced, about 1 million genetic markers initially was available.
Right.
But now more number of markers have been added. So those are the one which shows variations, or otherwise we call that as a polymorphism across different ethnic population.
Okay.
So those are the one we used to use earlier for understanding population history. But now the whole genome itself is available.
So in present studies, they usually how it is done in India now. It is with the entire genome sequencing.
So that is depending upon how much of resource we have,
See.
right? And also the output of talking about a particular population using only Y or using only mitochondria or genome-wide markers or whole genome, the outcome of the study will vary.
Okay.
Right? When you use Y chromosome, as I already mentioned, can look at only the paternal lineage. Mitochondria, only maternal lineage. When you use genome-wide association, you can also look at little wider perspective of both the pair. If you use whole genome, then you will get more information about the health perspective of the individual or of the population. If you use more, can understand the founder mutation and so on, particularly the recessive mutation, which will cause disease only when it exists in the homozygous condition or the copy of the chromosome. When we look at the population level, although today population may not have the disease because the mutation may exist in the heterozygous condition, and you can predict that when this mutation becomes homozygous, the individual
Will express that.
they have the disease. So that kind of thing one can predict by looking at the above.
Sir, coming to the health aspect, because you have, I think, one of the publication which is regarding to the heart attacks. So for that, early, like these days, we come in the news, we see, at a heart attack at a much younger age. So can these studies, genetic studies, help to identify that this person is predisposed to having a heart attacks with the mutation studies?
Yeah, not only heart attack,
Right.
any disease can be predicted, but one only issue is that not all the mutation which you have is going to cause the disease. If it is a single gene disorders, you know that if there is a mutation going to lead to the disease. Correct. Whereas the disease which are caused by multiple genes, multiple mutations, you can really predict because there could be something else which might suppress. Therefore, there's no disease, right? But it would actually help in condition where there's a well proved mutation and an individual. Wherever there's a mutation, 100% is going to cause the disease. In that scenario, some of the diseases, including the cardiomyopathy, can be delayed. That's what when we discuss with our clinical collaborators.
Yeah.
They say that you have the mutation because the cardiac phenotype, particularly hypertrophy or dilation, will happen after maybe 20 years, 30 years, 40 years, something like that, based on the type of mutation.
Correct. Right.
But when you look at very early age, for example, you sequence, there's a family, either father or mother, has cardiomyopathy, diagnosed at the age of 45 or 50. Right. Now that mutation is in the child who is only 20, for example. Right. Then you predict that the child, a son or daughter, going to have the disease up to 40 years. Right. But if you start treatment at very early age, as soon as you found that mutation, 20 years.
Right. I did a lot of buffer time to know.
Correct. If you start treatment, then you can delay the phenotype. Therefore, the individual will not have the disease. But even 10 years if you delay, yes, instead of 40 years, the person will get 50 years only get the disease. Or if you keep treating, you may even delay further. Right. So that kind of advantage is it come from the genetic studies.
Right. So not only the gene, sir, if there is a mutation, how can it treat it? Like on a clinical perspective,
There are
looking of.
multiple way of looking at. One, in broadly, if you look at genetic mutations, you cannot actually cure fully unless otherwise you do gene editing or scaling, which is not very common in India, but although there was successful, including cardiomyopathy. Okay. One can do that. But prevention is also equally important. Okay. Preventing the phenotype.
Right.
Okay. There are many diseases can be controlled by treatment. Sure, particularly for mitochondrial diseases. So, based on what kind of amino acid is being abnormal when the particular mutation is there. So, that the clinician treat with that. Similarly, Q coenzyme Q10 is used for treating some other mitochondrial disease. Okay, all right. So those will help to certain condition, not always.
Always, yeah. In general, sir, I'll try to if I make it very simple, because my idea is science should be for everybody.
Correct.
So in that way, if I have a gene and in that I take out there is a mutation which I've identified, which is you're saying as a marker for a disease which is cardiomyopathy.
Correct.
And that because of this mutation, the sequence is changed,
Correct.
right? ATGC sequence has changed.
Yeah.
And because of the sequence, the protein which is being formed, that is also getting changed.
Correct.
And that protein is creating a problem. So grossly, I'm telling it in a very layman language.
Sure.
So if I identify this chain, then I can know the pathway and then I can try to treat it. That is what you mean to say, right?
Yeah, correct.
Okay. But impact on cardiomyopathy is one. But again, I think you also worked on male infertility, osteoarthritis. There are a lot of other diseases can also be treated like this process.
Yeah. In general, genetic disease is very difficult to treat, right? For example, in the infertility, somebody is azoospermic. We can't really help because there's no sperm at all in the ejaculate. So what people do, again, use the available technology to aspirate and take out the germ cells and use ICSI technique.
Correct.
to fertilize. So that way people use it. But the treatment part is something very.
Yeah, I think that goes to the clinical side also. That is even not your area to work.
Correct.
So I'll just change to the other aspect of it. Is if how consanguinity is practiced in Indian communities, right? And what does it have health effects because of this practice?
Absolutely. So that's one of the major issues which we are seeing. And also, my research is at present towards that side. So, consanguinity is one which everybody knows that marrying within the family can lead to a disease. Other one is endogamy. Endogamy means marrying within the community. That I can say that it's an extended consanguinity.
True, true,
Right.
true.
So, what we are seeing now, not only the consanguinity and also the endogamy is also causing larger number of recessive disease. Recessive in the sense when both the copy of the chromosomes carry the mutation, only then it causes the disease. There are several such examples we found in our study. So, in fact, we predicted that at least 1/3 of the Indian populations are expected to have such a disease, large number, because genetically we looked at different individuals from the same community. They all shared a common ancestor, is same haplotype, right? So that gives a clue that when the recessive mutation happens somewhere in one of the chromosomes in a coding region, which ultimately is going to affect the protein synthesis. The protein is either truncated, right, or modified. Ultimately, is going to change the structure of the protein. Therefore, there's a disease. So when we looked at about 2,800 individuals, not only in India, also other South Asian countries, who belongs to 275 populations, we found at least 1/3 of the populations having very high IBD score, identity by descent. That means more individual from your particular community share a common haplotype. Okay, if there's a mutation which happened several generation back, that is going to be circulating because of the endogamy in a particular population.
Okay.
Today, a boy or girl carrying mutation one of the pairs, we call it as heterozygous. Yes, we absolutely normal. Okay, for example, it arose 10th generation back, and he or she marries somebody within the community, passing it to the next generation, then passing to the next generation. So, because they are marrying within the community, the frequency of the mutation, although they are normal, but their carrier is increasing.
Yeah.
After few generation, what will happen? We'll have high frequency of the mutation. Yes. Then the two individual, boy and girl, who are absolutely fine today, if they are carrying mutation heterozygous condition, if they marry, there's a 25% of chance that they contribute the abnormal chromosomes to the next generation, both, right? And that father homozygous condition become homozygous, therefore there's a disease. So we have identified large number of such communities in India. We are working on it.
Okay. So, but in India, this is a very big, I would say, as a social norm that marriages has to happen within the certain communities. Yes, it's a big thing when it doesn't happen within communities. Yes. So, in a way, knowingly or unknowingly, I'm not putting that, but this process or this, what is a culture is actually, encouraging to have more frequencies of this heterozygous conditions.
Yes, absolutely.
Leading into a chances of more disease.
Correct. So that's one of the reasons that why India has large number of such recessive disease or rare disease. There are about 70 million people affected with the rare diseases. So our prediction is 1/3 of the populations are expected to have a population specific rare disease. And even our own experience, what we have seen is these kind of mutation lead to disease which is lethal at very, very early age. The children with the mutation, homozygous mutation, die at very, very early age. So even today, if you go to any population which is very small in size, because of the endogamy, the children die at very early age. Where you go to a particular community, you may not see any individual with the disease because the children with the homozygous mutation has the disease and they die at very early age, less than few months after the birth. So superficially, if you look at, there is no disease, whereas most of the individuals in a particular community carrying the mutation, which is potentially going to cause the disease when it exists in the homozygous condition.
Correct. I think the classical example for that is thalassemia.
Thalassemia is one, but even uh, more than that, also we have, yes.
Right.
There are some disease which we are working on it.
Because I come from the eastern part of the country, where thalassemia is very predominant. And I can totally correlate with this because in society you will not find anybody suffering from thalassemia. At very early age is diagnosed and the survival rate is hardly max to max 18 to 20 years.
So at least they are 18 to 20 years. In some cases, children die within a few months after the birth. Right? It's not going to happen to every child. But it's going to happen the child with the homozygous mutation. That means some child are fine, they're normal. Some may carrier, but absolutely fine. But some with homozygous mutation, they died. That means in a given point of time, you go to a community, would be absolutely fine. Correct. So we are more worried about that kind of population. So try to, even some cases, we identified, we are screening the entire population with the idea to identify those who are carrier, even children who are carrying the mutation. We wanted to make a database, want to find somebody in the population itself to take it for a very long period. Today, a boy or girl, they're going to school, maybe 10 years now. Right. After 15 years, when they are ready to marry, this data will be useful. Correct. Right. Correct. So they can match, either boy want to marry or girl want to marry. So they can match with the apparent individual. Correct. And we can counsel them. Therefore, this disease can be avoided in the future generations.
So that is where I think genetic counseling is very, very important.
Correct.
When marriages and these are
Yes,
alliances are happening.
very, very important. Particularly when you have disease like that. People want to marry within the community. They don't want to go outside the community. Who are carrying the mutation identified, and unless otherwise you do this way, we are not going to prevent the disease.
Correct.
In preventing this is two way. One is the treatment. Treatment will help only in the case where you can at least manage the disease. On the other side, this kind of disease which actually life threatening at very early age, you cannot treat. You have to avoid. So for genetic disease, rather than treatment, I think avoiding is the best option.
I'll just reminded you, sir. I had a colleague, so we were, he's from AIMS Delhi, did all the prenatal testing of the child to be yet to be born. And out of all the parameters, everything looked okay. After doing that, when his daughter was born, she was diagnosed with Down syndrome. And Down syndrome is like very easily identified by seeing it. But out of all the parameters, there was a one or two parameters that might minor changes.
Right.
Even the doctors could not predict it. But when she is born, there's no other way you have to manage. That's
Correct. Correct. Correct.
so that was a being in a hospital setup also. This happens to an individual. Yes. Imagine in a community who doesn't have any awareness about it.
Absolutely. Absolutely. Yeah. So the Down syndrome, these are chromosomal abnormalities, and in this case, probably they must have looked at the common disease which comes. They must have screened only for those markers, and they must have totally ignored the chromosomal aberrations. This would have been very much easier to look at taking amniotic fluid and see whether there's one more chromosome 21 exists, or even the other common disease like Turner syndrome, Klein-Perther also would have been checked. But people may not have no thought that it would happen, because that is a very, very rare chance that because we cannot predict the Down syndrome uh, because of the non-distinction of the chromosome while chromosome cell division, it happens.
This is a very interesting topic. And in India, many people are even not aware till date. Yes. Why I'm saying this? Because, no, well, I went through when my child was more to be born. That time also we saw a lot of checkpoints. We made sure, nah. But grossly, doctors, as you rightly said, no, they will see major problems and then they will say, okay, everything looks good.
Correct.
Two things what I feel is this testing should be little cheaper so that every person should go through it.
Yeah.
And there should be a standard which God ICMR always has. But in practice also, like people should be doing, okay, these are the checkpoints, they should be doing it.
Correct.
To be assured that everything genetically, whoever the child is coming on this planet,
Correct.
they are genetically safe and sound. That is one point. So moving on to a little different is on the forensic side, because genetics again, they're also for like crime, disputed parents or disputed child,
Yeah.
inheritance. Their genetic plays a very important role, right?
Yes.
So in forensic sciences, grossly, if I see in crime scenes, how genetics can be applied to come to a suspect or a criminal identification. I'm targeting a larger area. Sorry for that.
No.
But this is a very interesting topic again.
Yes. Genetics actually play a major role in forensic, right? Again, the same principle. So every individual inherit of a genetic material from father, of a genetic material from the mother.
Yeah.
For simple paternity or maternity, to identifying any major crime investigation, genetics play a major, right?
Yeah.
Because of the transmission. For example, paternity, maternity is a very simple thing.
Yeah.
We can identify. That is identified based on the repeated sequence which is present in the chromosomes.
Yeah.
Right. Now there are standardized markers are available. If you take a pair of chromosome, for example, chromosome 1, right? We inherit one chromosome of the pair, chromosome 1. Yeah. We talk about one from the mother, one from the father. We look at a specific region on the chromosome. Yeah. That region, we are looking at the repetitive sequence.
Correct.
Father's chromosome might probably have 8 repeat. Mothers might have 10 repeat, for example.
Yeah.
Okay. Then you expect the child will have 8 repeat from the father, 10 repeat from the mother. Although there's another chromosome which I am not on. Right. Similarly, if you look at multiple such region, if you analyze the child, it could be the male or female, then you will have the pattern for every locus, every region of the chromosome, wherever there's a repetition. If you could match one chromosome repeat with the father, one with the mother, then you can say that yes, these two are the parents of this particular child.
So this is a biological son of this two
Correct.
individuals.
Yeah, absolutely. Yeah, you can able to identify. This is in a simple paternity or maternity cases. Same way for any crime investigation. Wherever there's a crime happen, there could be some biological material left by the culprit. Right? It could be a blood, it could be a saliva, it could be air, right? So any biological, it could be a semen, so on. We can able to isolate DNA from that small biological sample found in the crime scene. We can identify the person who is involved that crime. Even the person, I can go little wider than what is known. If you use, for example, Y chromosome and mitochondrial DNA. So what earlier I said was the nuclear genome where we have two copies. Correct. In a old setup, for example, the crime happened 10 years ago.
Oh, okay.
Or even
Before that.
before that, you can able to sequence, uh, if you could able to sequence the complete genome of the individual, that's well and good. Otherwise, we can amplify the mitochondrial DNA, sequence them. You can also amplify the Y chromosome and do that. If you are unable to use the nucleation, then you can trace the paternal and maternal lineages and try to identify who is the person.
Okay. Okay.
Even today, you may not see any sign of that individual, but it can actually trace and find out, yes, this person belongs to this particular family, provided you have sample of that family, or even also broadly say based on the haplogroup. For example, in the mitochondria and Y chromosome, based on the mutation present in different region of the mitochondrial DNA, but it was 60,000 sequence. 16,000 kb is the mitochondrial DNA. If you sequence, you are able to identify the mutation different position. Based on that, we can assign a particular haplogroup to every single mitochondrial DNA. Correct. Right. If you look at the mitochondrial tree, there's a L is the founder for the mitochondrial haplogroup. Most is found in Africa. Then there is a two branch, M and N. M is found broadly in India. Right. Similarly, as the individual acquire more mutation, they fall into different haplogroups. Right. And if you look at the distribution of mitochondrial haplogroup, the worldwide, you can clearly see the original African, most of them will have L haplogroup. Majority of the Indian have M haplogroup, and in the East Asian have N and B haplogroup. Similarly, you can see different continent have different kind of haplogroup. Same is true for the Y chromosome also. So even today, you don't see anybody, but broadly you can say, if you found L haplogroup, you can say probably this person belongs to Africa. This, yeah, right. So it's amazing. If you broadly found M haplogroup, although there are several sub haplogroup, but broadly if you look at M, you can say from India or South Asia. Correct. And I correct. So that much one can able to predict by using the Y chromosome and the mitochondria. Right. And now it's also possible to use some of the phenotypic markers. For example, if you look at the mutation which is responsible for the blue eye, those are very present and only restricted. Yes, you can say, oh, this person might belongs to that region. Yeah, right. Even for the curly hair, color of the hair, there are markers which determines that, including cardiomyopathy, and you can say that because that's not present outside India or outside South Asia. If you find the marker 25 base pair deletion in an individual, you can say proudly they are somewhere in South Asia. Right, right. So right. Obviously in the entire globe, you have just a small area. Correct. Or work becomes, you can say, yes, here. Even if you don't have precisely, but you can broadly say that this person might belongs to this continent or this subcontinent or this country. You can go to that level.
But so, if we see our like paleontologists, they have this, fossils and bones. So from there, how long back, like if I have a bone or a teeth collected, how long back can we take samples? Like, is it like 6,000 years or so? And from there, DNA can be isolated to do these studies?
Yeah, absolutely. Uh, if the biological material is preserved very well, one can isolate DNA from several thousands of year old sample. Well, we are part of one of the major studies in Europe. In fact, we traced the origin of Europeans. There were three different places which are of 7,000 and 8,000 year old bone sample in Luxembourg and many other places. Even much older, you must have seen Savante Paber's work. He looked at Neanderthals, which is of more than 500,000 year old. Right? So one can able to isolate as long as it is preserved very well. But unfortunately, we don't have such biological preservation in India because of the climatic. Yes.
The people say, when the Arctic poles or the snow caps melt, there might be a lot of
No, because ours is tropical country where no hot, humid, and so on. So bone sample are not preserved very well. Yeah. So one successful thing is 1,500-year-old sample which we analyze from Rupkund. Yes, this is about the place is about 5,000 meter above the sea level. So all the time the well is the lake is frozen condition. Therefore, we could able to go. Other successful one is the Rakigiri sample, which is about 3,000 year old sample. It was a successful story again with collaboration with many groups. We could able to do that.
right. So, thank you for sharing all these informations and making it so look simple. I know none of this is simple. But again, if you see your journey, any aspect of like if you get time, like any aspect you would like to work in future, or any area which you thought if I had time I would have done that.
There is no regret that I could not do this or that.
Yeah.
So whatever I felt, it's very important and interesting, very unique to India. So I've been working on it. Like for example, the understanding population history is very very interesting. Because not only the biologists, not only the scientists, even the common people is very interested in knowing about the history of human population. So that's one thing which everybody like it. Similarly, understanding our health and disease is also very, very important. Okay. Fortunately, because I could work on both aspects, I could also mix both for better understanding of our health and disease. Because we know the history, so we know the disease.
Absolutely.
Then we connect because the population history helped us in better understanding of some of the rare diseases. Why rare diseases are very common, how it actually happened. We could predict using a genetic tool, and we can actually look at, because we don't know some of the populations whether they have the disease or not. By looking at the genetics, we went back to the population, and in fact we found the disease. So yeah,
That is where genetics is sitting at that junction.
correct.
It has the power to see the past
Yes.
and then also predict the future.
Correct.
That is, I think, one of the rarest tool available to us.
Yes. Yeah, so that one point which you brought, by looking at the ancient sample, which are of several thousands of years old,
Yeah.
we can actually see how the DNA was, for example, 7,000, 8,000 years back. We can again correlate with the genetic evolution, our adaptation, and so on. For example, the 8,000 year old sample, which I was saying, when we look at the human history, how the agriculture started, how the domestication of animals started, all we know, right? We can actually connect how our genome has evolved based on the agriculture, domestication of animals, and so on. So the agriculture started about 10, 11,000 years back. Right? The domestication of animal and starting drinking of the milk started much later, around 4 to 5,000 years. Yes. But if we look at the DNA of 7, 8,000 year old, we can actually correlate with our culture and the history. If you see, because it's 7,000, 8,000 year old, we all gained the mutation After domestication, started drinking the milk, we gained a mutation lactate persistent gene for digesting the milk sugar very effectively. Okay, otherwise, we all have very active enzyme when we are at the young age. While drinking the milk, after that we lose that, and therefore we cannot digest the milk sugar, and we face a lot of other issues, including stomach ache and many other. Even today, some of them cannot tolerate.
Yeah,
the milk.
tolerance is very predominant
Yes.
these days. Yeah.
Correct, right? So when we look at the mutation, so they have not actually have the mutation. Some of the bone sample which are of 7,000-8,000 year old, because this arose very recently,
Right. Right.
right?
So that is because of the practice
Yes,
that mutation had to happen
correct,
so that we evolve to digest milk.
correct. Same way when we started eating rice and starch rich food, the amylase gene copy number increased, right?
Right.
Same way the skin color mutation, which we talked about, some regions dark, some regions, because that is because of the adaptation. When modern humans started migrating towards Europe, they might have faced a lot of issues because of the vitamin D, lack of vitamin D. For that we need sunlight. The Europe, even some places, even months together, there's no sunlight, right? To overcome that, there's a mutation introduced in a gene called SLC24 25A that made skin color into dark into light skin color. Therefore, with the mild light, people could be able to synthesize vitamin D. That's how the mutation arose. That we see predominantly in Europe, but not in India. In India,
It was a tropical climate.
correct. So that is required because of survival of humans, right?
Yeah, this is, I think that is how the nature's beauty comes
Correct.
into picture.
Yeah, even sickle cell, if you take, because malaria prevalent place to prevent malaria parasite entering into a blood cell. So that mutation arose, but of course it causes different kind of problem to
Capacity and all.
correct.
But again, sir, I think interestingly, when you mentioned this, the northeastern populations have a different type of HB.
Correct.
That's what I was working on that time. So HBE variant doesn't seem to have effect with the plasmodial load.
Correct.
So that is something. Yeah.
Yeah.
So they have learned to coexist
Yeah.
together.
Correct. So that adaptation, naturally adapted, um, like some of the example which I was saying. So because of something which we eat, to tolerate that, there's adaptation. So multiple ways there was adaptation. They're all slowly evolving. That can be seen by looking at the ancient year.
Correct. Correct. Yes. Yes. So when you have the first copy, first draft, you can find out how many corrections have happened.
Correct.
Yeah, that's true. That's a very nice way of saying it. Even sir, like we were discussing, the pollutions and those things are increasing day by day. Does it also have health effects? Obviously, it has on the functionality. But over a period of time, will we get adapted to this environment and then we'll try to perform better?
Yeah. Because if you are in adverse environment, naturally we try to adapt to that environment. Right? For example, those who are living in high altitude, because the oxygen level is very low, and we need oxygen. So there is again the mutation introduced. Therefore, the RBC carry more oxygen. Right? That's the way they adopted there. And that we don't have that kind of adaptation here. And when we are exposed ourselves in a environment where there is a pathogen, where there is a pollution, so we try to overcome that problem. Initially, probably we may face some problem, but for long exposure, to give better advantage to survive in that particular place for a very long time. Yeah, right. So that even we can see, I can take my own example because I born and brought up in the village, right? So until maybe move to higher education or something, I've been exposed to in a village setup, right? We sort of feel all kind of no exposure, but probably I feel So that would have given me much more immune thing compared to those who are living in very sophisticated place, drinking the filter water, no, right from the childhood.
Yes, agreed, sir. I agreed.
And they're not exposed to this kind of environment. And that aspect, for example, if you look at the Andaman tribes, who are in very, very close environment, and they have been selected to, naturally selected to live in that particular environment.
Okay.
And if they are exposed to the environment in which we are there today, they're not able to survive in much better way.
I agree. Totally agree.
So, I'm just giving one example. Say that there are many people. So the slow adaptation gives better survival for that particular period. Even if you take other common diseases like diabetes, our forefathers, they did not have much food, right? So they must have taken whatever the limited food, and our genome is adapted to that food, right? Now we are exposed to within, I think, 2-3 generation.
Yes, yes. Overeating and all.
Correct.
Yeah.
And not only overeating, and also the richness of the food.
Hmm.
Our genome is adapted for low metabolizing with the limited food. But now we are giving all kind of food. That's where all the diabetes, cardiovascular, lifestyle diseases are coming.
Correct.
If this happens for a long time, I'm sure that there will be some mutation which can overcome,
Yeah.
but we don't know when it will happen. Right? These are all only the predictions,
It's only better.
so that we can have our generation, next generation, can have
True.
overcome all this.
So yeah, that's true. I was just discussing with one of the professor names. They were saying that over a period of time, our body parts like hands, you see, they have great functionality.
Correct.
But thumb is the most used. You use it for your phone.
Correct.
So after a certain time, your thumb might evolve better. Rest of the fingers might not.
Well,
So you said, hey, it's possible. It's how you use and disuse.
correct.
Right, so it was absolute pleasure. I have no words to actually thank you for this wonderful experience, and I'm pretty sure whoever will be watching this podcast, they will definitely be immensely, getting. I have spent almost like roughly 1 hour with you, and I've tried to get whatever I could so that they get experience out of that. But I don't know, 1 hour is like nothing. So thank you for agreeing to come here on this platform and then give your time. Immensely thank you, sir.
Thank you. Thank you so much for coming over here. And I'm really happy that, so whatever you do in the lab actually has to go to the common people. They should understand. So that way, I think you are doing great job to taking science to a common people. So wish you all the very best. And I wish you for many more such interview with others who are doing excellent work, and take that to a common people. I wish you all the very best.
Thank you.
Thank you so much.
Coming from you means a lot. Thank you.